Hello and welcome. Today we're going to talk about meiosis and inheritance. You might even want to break it up into two parts. It's a lot of information, but they're so closely related that I want to put them together. So here we have two chromosomes, and both, if you notice, are in this X form. So that really means we have a sister chromatid here, a sister chromatid here, which are exact copies of each other. And then we have a sister chromatid here and a sister chromatid here, which means those two are exact copies of each other. These two chromosomes are homologous, all right, which means they have the same sorts of information but might have different versions. For example, this one might be blood type A, and this one might be for blood type O. So we're going to look at how those characteristics get passed down to the next level, and that's going to happen through this process of meiosis. So I've linked Mr. Anderson's video into the Google assignment so that you can watch it, and he gets the credit for it, not me. That would be very wrong of me to record this and show it on mine. So we'll move on. Make sure you watch that. So what you should have learned from Mr. Anderson's video is that the main point, the whole reason we go through meiosis, is so we can have sexual reproduction, which creates variation. And there's three big ways that it creates variation. We get variation in prophase through synapsis and crossover. We get a ton of variation with how these can line up randomly. And then lastly, the result, the fertilization, that also leads to variation occurring. So here we see kind of that first example of how the random way these chromosomes line up in homologous pairs leads to variation. And homologous chromosomes, just like I said at the beginning, this pair, they're homo, they're same, right? Homo means same. They are the same sorts of information, but may have different versions. So this one might have a gene for blonde hair, and this one might have a gene for brown hair. So the same trait, but different allele. Another vocab word that's talked about is tetrad. So tetra means four. Just think like Tetris. So we have 1, 2, 3, 4 sister chromatids. And then this whole thing, the lining up randomly, that's independent assortment. Here we have our crossover happening. We've traded pieces of these chromosomes. That's adding to the genetic variation. And this also helps us, we call this recombination, actually helps us figure out which genes are linked, which genes aren't linked, and then even how close they are, where they are on this chromosome. So we had Henry Morgan kind of figure this idea of linked genes out with the white-eyed fruit flies. And we can actually use that recombination rate to predict exactly where on the chromosome that particular allele is, that particular gene. This all happens during prophase 1, crossover happens. And then this last form of adding genetic variation has to do with which of the, you make millions of sperm and one egg, and kind of goes back to what Mr. Anderson was saying before with the independent assortment. You have a 1 in 8 million chance which way they're going to line up for the sperm, a 1 in 8 million chance how they're going to line up for the egg that gets fertilized. That's a 1 in 64 trillion. And then here we're going over kind of two big important vocabulary words, diploid versus haploid, and somatic cell versus gamete. So haploid is when we have just the single chromosome from just mom or dad. That's what we're seeing in your sperm and egg, which we call gametes. Pollen would be another version of a gamete. And then diploid, di means two. You have two copies, one from mom, one from dad. And so that happens in our somatic cells. We're diploid. So a skin cell, a muscle cell, a cell in your intestine, they're all going to be diploid. And remember what we said before, all your cells have the complete set, so they can make any type of cell, but once they've been specialized, once they've been differentiated, they're only going to make one type of cell. Now, How's the function of haploid related to why it has 23 chromosomes and not 24, not 46? This is my sperm cell or egg cell. It has half the amount. So when I put half with half, when I put 23 with 23, those combine to make 46. If I had a 46 chromosome sperm and a 46 chromosome egg, that's going to come together and make 92. That's double the amount. That's polyploidy. That's a problem. Like you have bananas. One of the nice things about bananas is it doesn't have seeds. You can't take that banana and plant it because they are polyploidy. They have to clone those. So when you have polyploidy, that leads to gene expression problems. Often sterility is one of those, like we see in bananas. Here's another video you should watch that kind of goes over how Mendel helped us understand how genes go together. Again, it's a limited understanding. Mendel figured out some ways that worked with pea plants for specific genes, but that's not the whole story. But it really was important because it led us to really understanding and being able to start seeing that some of this is predictable. So two big important words, genotype and phenotype. Genotype is those A's, T's, C's, and G's, all right? Thinking about what the gene actually says. Phenotype is what actually gets produced from that gene. So when we look at this, oh, this looks familiar from 7th grade. This is that Punnett square. We have the big B, which is dominant, the little b, which is recessive. We think of this as our genotype. This is what the DNA is coding for. And little b, that's our genotype as well. The phenotype is what actually gets expressed. So what does this mean? So let's say I'm talking about hitchhiker's thumb. My thumb goes back, all right? That's a dominant characteristic. So dad here has hitchhiker's thumb. Mom has just a straight thumb. She can't make it go back. She just has it go like that. So if they are having offspring together, this is saying there's a 2 out of 4 or 50% chance that they'll have a hitchhiker kid and a 50% chance that they will have a straight thumb, not hitchhiker thumb kid. So phenotype, again, I kind of got into that already, is what gets expressed. One thing that I don't like is a lot of times we think about is what you can see, but the liver cell, how that cell develops, that's the phenotype, but you don't really see that. So all those proteins that get made, everything that makes you, that's the phenotype, and it's what gets produced. That's how I like to think of it. So one thing, just like we talked about Mendel's peas earlier, is was it a yellow pea or was it a green pea? There's two possibilities. Was it a short plant or a tall plant? There's two possibilities. It's not as simple as that for everything. There's, for most, a whole spectrum of what gets expressed. So, how are phenotypes related to genotypes? The key here is, the thing that I'd write down is that genotypes determine the phenotype. Genotype codes for the phenotype. Alleles are different versions of each particular gene. We've talked about alleles before, so it's not brand new. That's really important to keep going over the same thing. So my big B and little b, those are what's representing my allele. What version of the trait? So I had talked about hitchhiker thumb before. What type of thumb do you have? Do you have a straight thumb allele or do you have a hitchhiker allele? And it takes two to kind of determine that. So if you have one big B, you're going to have that hitchhiker thumb. If you have one little b, doesn't matter. The only way you can show a straight thumb is if you have two little b's. Locus is another little vocab word that just means where on the chromosome it's located. All right. So again, it says alleles are often represented by letters. Here we're talking big B, little b. The actual letter that you choose doesn't matter. I prefer to use ones that are easy to tell between the capital and lowercase. An example of one that's a bad one to use would be like, now I can't think, but like V, for example, would be a bad one because it just looks like a smaller version of the same thing. this, again, how to draw a Punnett square is going to go over how to actually use that Punnett square. Watch that as a good way to review. Here we go. The law of dominance. There's 3 of Mendel's laws. The first one is the law of dominance. And that's where I'm talking about you have to have 2 little r's in order to see the recessive. But if you have any of the dominant r's, you're always going to see that dominant phenotype. So this one's red eyes, that one's red eyes, that one's red eyes, this one's sepia. Let's move on. Why am I not? That must mean I'm the last slide. So that's kind of it for the basics of inheritance. Again, kind of bringing anything together. Where did these big R's and little r's come from? Why is it random which ones we get? That all has to do with meiosis. So these alleles that we get, that's the randomness that comes from meiosis. Right? So that's that. Take your time to read back. Feel free to go back, pause, write down some notes, highlight, write some questions, those sorts of things. Hope that was a little bit helpful for you. All right, now I just need to figure out how to stop.